Biofertilizers are biological substances containing living microorganisms such as bacteria, mycorrhizal fungi and cyanobacteria, which when added to the soil increase its fertility and promote the growth of plants and trees. Mycorrhizal fungi preferentially extract minerals from soil organic matter for plants. Bacteria fix atmospheric nitrogen into biological usable form. They also help in solubilizing phosphorus and nitrogen. In addition, cyanobacteria, formerly known as blue-green algae, also fix atmospheric nitrogen. Hello and welcome to my channel, Sam Biology Class. Friends, today we will discuss about one of the biofertilizer known as azospirillum. Azospirillum is a gram-negative microaerophilic nitrogen-fixing bacterial genus. It belongs to the family Rhodospirillacea. It can promote plant growth. Azospirillum is gram-negative and slightly twisted oblong rod-shaped. It has at least one flagellum, but sometimes they also use multiple flagella to move rapidly. Azospirillum is aerobic, but under low oxygen conditions, some of them also function as microaerobic diazotrophs, means when the oxygen level is low, they fix atmospheric nitrogen into ammonia. In addition, few species are facultative anaerobes. That means, if necessary, they can live without oxygen. Azospirillum is found in freshwater and soil habitat where it maintains close relationship with plant roots. This relationship, known as associative symbiosis, is largely beneficial. More than 113 plant species from 35 different plant families have been documented to have benefited from this relationship. Now the question is, what is associative symbiosis? It is a loose, non-obligatory ecological relationship where the microbe colonizes the space around the plant body or roots, mutually exchanging benefits. But in this relationship, specialized enclosed structures such as root nodules are not formed. Azospirillum is used as a biofertilizer. It promotes plant growth through a variety of mechanisms. Number 1, some azospirillum species excrete plant hormones which alter the growth of plant roots. Affected roots grow more branches and fine root hairs, thereby helping the plants to acquire more nutrients and water from the soil. Number 2, it alters the form of nitrogen and phosphorus in soil and making them more available to the plants. Number 3, azospirillum produces antioxidants which protects the plant from stress due to drought and flooding. In addition, azospirillum also promotes the growth of plants indirectly. It reduces plant disease by competing with pathogen on roots for space and trace elements such as iron. Isolation of azospirillum. First step is sample collection. Root samples are collected from the rhizosphere of sugarcane, grasses or cereals. Second step is surface sterilization, for which the collected root samples are surface sterilized with 0.1% mercury chloride or 70% ethanol in order to remove the surface contaminants. Third step is enrichment. In this step, the root pieces are transferred to a nitrogen-free malate containing medium which is highly selective. Enrichment is followed by incubation at 30 degrees centigrade or 37 degrees centigrade, which create a distinct pellicle, white pellicle, below the surface. Last step is pure culture isolation. For this, the white pellicle, which is formed below the surface, is streaked on Congo agar red medium, and the microbe is characterized by the scarlet red pigmentation of colonies. After isolation of pure culture, it is important to identify the microbe, which is Azospirillum. For identification, different methods are available. One is microscopical examination, second one is biochemical test, and third one is appearance. Microscopically, Azospirillum can be identified based on their shape. The microbe is gram-negative. Its shape is spiral or vibroid motile rods containing polyhydroxybutyrate granules. The presence of polyhydroxybutyrate granules indicate the presence of Azospirillum. Second test is biochemical test. Azospirillum can be identified based on their ability to grow efficiently on media containing malate, succinate, lactate and pyruvate, but not on glucose. Third test is based on appearance. Azospirillum can be identified based on their distinctive white or pinkish wrinkled colonies. So next we will come to know about the process for the production of mass inoculum and its field application. Now the question is how to prepare the mass inoculum of Azospirillum. It is achieved by first preparing the starter culture by transferring a loop full of Azospirillum colonies to a culture flask having ammonium containing Ocon's medium. The flask is then incubated under aerobic condition at 35 degrees centigrade on a shaker incubator. Under this condition, Azospirillum proliferates. In next step, the cells are harvested and are transferred to a fermenter for further growth by maintaining aerobic condition at 35 degrees centigrade for few days. The broth is harvested when the cell number reaches 10 to power 9 per ml. Next, we will know about field application. To apply azospirillum as a biofertilizer on the field, different methods are available. First one is seed treatment. In this method, slurry of azospirillum is prepared by mixing suitable amount of culture with water. Then the seeds to be sown on the field are soaked in the slurry for a suitable time period, and then the seeds are sown on the field. Second method is seedling treatment. In this method also the inoculum slurry is prepared, and then the roots of the seedlings to be transplanted in the field are dipped in the slurry for few minutes, following which the seedlings are transplanted in the field and remaining slurry is spread throughout the field. Last one is top dressing. In this method, carrier-based inoculants is prepared by mixing the microbe with soil and manure in suitable proportion, and then the mixture is top dressed in the field, particularly around the seedlings. So friends, this is all about isolation, identification, production of mass inoculum and field application of azospirillum. For more updates, subscribe my YouTube channel, Sam Biology Class.